In this video, we're going to be talking about Mendel and some genetics fundamentals, or some fundamental principles of genetics. So, Gregor Mendel, he's often considered to be the father of genetics. He was an Austrian monk who studied botany. And when he studied botany, his main subject for his experiments were garden peas. And so the reason why he chose garden peas was because they were immobile. So they're not going to move around and interbreed with each other necessarily. They're stationary. They don't really take up that much space, and they have a very fast turnover, meaning you could mate two different plants with each other, and you're going to get results for your experiment pretty quickly because they're quick growing, and they're annual, so they seed and flower yearly. And then lastly, well, not lastly, but another reason is because they are insect pollinated rather than wind pollinated, meaning he had more control over what plant gets to mate with what plant. There's less room for error if they are evolved to receive pollen from plants by the means of pollinators rather than wind, which would mean that they would be very sensitive to any kind of pollen in the air. So the way you could hand pollinate them potentially is what you would do is with, one of your pea plants, you would remove the male, the pollen producing organ, the anthers. So you'd remove the anthers by cutting their filaments, leaving only the female part, which is the stigma. And then from the other plant, so you have two pea plants, it takes two to tango. So the first pea plant, you remove the male part, and then in the second plant, what you do is you extract some pollen from the second pea plant, and then you pollinate that first pea plant with the pollen. So you rub some of that pollen on the stigma, and the reason why you cut off the anthers from the first one is so that it doesn't self-pollinate. So he's pretty much forcing, the cross between two plants. All right, so yeah, another main reason why he picked them is because they're easy to hand pollinate. So it gives him a lot of control over what gets to mate with what. Now, when looking at these pea plants, he found that they had this tendency to pass on traits that were kind of binary in nature, meaning there was no in between. So, for example, with flower color, you would have purple flowers or you'd have white flowers. You wouldn't have anything in between. There would be no intermediate, like light purple. Same with seed color, for example. You either had yellow seed color or green seed color, nothing in between. Same with seed shape. You'd have round seeds or you'd have wrinkled seeds. You wouldn't have like some roundish, wrinkledish seed intermediate phenotype. It would either be round or wrinkled. Pretty binary. So he studied 7 characteristics, each with 2 traits, one or the other. And then just as some terminology, true breeding just means purebred. That means over multiple generations, you're breeding for a particular trait in that organism. So when studying the pea plants, one of the theories that he wanted to disprove was irreversible blending theory. His question was, when two strains with contrasting traits breed, are their characteristics irreversibly blended in succeeding generations? So according to irreversible blending theory, we're going to use this example to explain it. Well, so irreversible blending theory would mean that, when you mesh two traits together that are the same, for example, spherical, so you have, one parent that has spherical seeds and the other parent that has spherical seeds, their offspring would all have spherical seeds. Okay, so irreversible blending theory just means that you cannot necessarily backtrack. And to use this as an example, so the P generation, which is the parental generation, what he had was a true breeding spherical seeded plant and a true breeding wrinkled seeded plant. And so he cross-pollinated those, and all of their offspring had spherical seeds. Okay, so that would imply that, maybe, the spherical seeds are probably that allele, the allele that contributes to spherical seeds is probably dominant, whereas the wrinkled seed is actually recessive. but he didn't even know that at the time. So all of the F1 generation, or the first filial generation plants, had spherical seeds. Now, according to irreversible blending theory, if he were to take one of these spherical seeded plants and mate it with another spherical seeded plant, the result would be 100% spherical seeded plants. Now, that's not necessarily what he found, but we'll see that in 2 slides from now. So this is the first part of his experiment. What he did was he took, true bred spherical mated with true bred wrinkled seeds. All of the offspring were spherical seeds. Now, before we jump further into the results of his experiment, let's just talk about what he or some of the terminology. So what he did was a monohybrid cross. So that's where one characteristic is looked at. I need to fix that. So one characteristic is looked at, and that's going to be seed shape in this case. So mono meaning one, a monohybrid cross. One characteristic is looked at, and that characteristic, seed shape, could have 2 different types. In this case, it was spherical or wrinkled. So the P generation is the parental generation. The filial, so any kind of F generation, is just offspring generation. So the first filial generation is the first set of offspring. So it's like, the P generation is the parental generation. They make the first filial generation, and then after the first filial generation, they're going to make the F2 generation, the second filial generation. So that'd be like the grandchildren, for example. So P, the parental generation. So the parental generation is true rounded, and he crossed it with true wrinkled seeds. And then the F generation, so the result, was all of them were round, none of them were wrinkled. Just 2 more terms here. If the allele is dominant, that just means that it masks the other trait. And then if an allele is recessive, that just means that when that allele is present, even though it's present, it's going to be masked by whatever dominant allele is present. So if you're in the heterozygote, you would therefore have a round seed, because your wrinkled allele, or the allele that contributes to the wrinkled phenotype, would be masked by the dominant allele. All right, so seed shape, you have either round or you have wrinkled. So let's look at the results one more time, and we actually go a step further here. So the seed shape, so the true bred smooth seeds were big S, and the true bred wrinkled seeds were little s. And so the parental generation, this is showing the monohybrid cross using a Punnett square. So you have big S, and you're making a cross between little s, and so what you get is a big S, little s, big S, little s, big S, little s, big S, little s. So all 4 of these teal boxes would be filled with big S, little s. All right, so this is going to be, all of these teal boxes are the first filial generation. So what he did was he took one of the first filial generation plants and he mated that plant with another plant of the first filial generation. So here, this is kind of showing it. You have big S, little s. These are their gametes that they're contributing, and all of your offspring are going to be big S, little s. And then what he does is he mates big S, little s with big S, little s. And these are just the gametes that they're contributing. You could have a little s gamete, or you could have a big S gamete, because they're heterozygotes. Hetero meaning different. So you have 2 different variants of the same letter in this sense. All right, so this is where it takes us a step further. So he mated the F1 generation with another F1 generation plant, and so now we get a monohybrid cross here, a big S, little s, mated with a big S, little s. And so if you do the monohybrid cross here, big S, the cross between big S and big S is 2 big S's. The cross between a big S and a little s is a big S and a little s. The cross between a little s and a little s makes a little s. And so in this case, in the F2 generation, he actually got wrinkled seeds, which disproves irreversible blending theory. So here's the F1 plant here. And what he did was he cross-pollinated. So this is just showing a cross-pollination or pollination of self. And he found that, 25% of the offspring had wrinkled seeds. So his conclusion was there's no irreversible blending of characteristics, and that the recessive trait, even though it was present, which he, at the time probably didn't necessarily uh, know about alleles and genes and DNA and the physiology of inheritance, or the chemical properties of inheritance. Um, they were carrying the recessive allele. 100% of the F1 generation were heterozygotes. However, they did not express the wrinkled phenotype because they had the dominant spherical allele present. And so when he cross-pollinated them, it wasn't until the F2 generation that you had a chance of even getting a homozygous recessive plant, which is the wrinkled seed. Homo meaning same, so homozygous meaning, little less, little less. All right, so Mendel's laws of particulate inheritance. According to particulate theory, there are alternative forms of particles or alleles that make a genotype. That just means that for your genes, there are variants of those genes, which are pretty much alleles. Alleles are variants of particular genes. Second tenet is an organism will inherit two alleles, one from each parent. So you could either be homozygous, meaning you have the same two alleles, or you're true breeding, or you're heterozygous for a specific gene. You're heterozygous for a specific gene, meaning you have two different alleles. Or you could be considered to be carriers of some kind of trait, not necessarily expressing it. All right, and then the third one is that if an allele is displayed in the phenotype in the physical characteristic, then it's considered dominant, and then if allele is masked, if it has the potential to be masked, then it's recessive. And then for a sperm or egg has only one allele for every gene because they separate by meiosis, or the law of segregation. So this is just pretty much saying that, your sperm is going to be haploid, your egg is going to be haploid, or 1N. When they fuse together, what they're doing is they're bringing, half of the genetic material from the one parent, and then the other one is going to bring the other half of its genetic material from the other parent, creating that zygote. So sperm and egg only has one allele for every gene. They're haploid. All right, alleles separate. So number 5, alleles separate independently from one another during meiosis. We call this the law of independent assortment. That's something that we cover in meiosis. And then here it's just it's prompting us to use a Punnett square to figure out the probability of offspring genotypes and phenotypes. We've already done a Punnett square or a monohybrid cross. So, and we're actually going to do a dihybrid cross too. So we're going to use a Punnett square. It'll just be a larger Punnett square because we're going to be looking at two different characteristics simultaneously. So Mendel didn't necessarily, at the time he was doing his experiments, he didn't really have knowledge of meiosis or of DNA. But he did do some dihybrid crosses, which would probably help one better understand the law of independent assortment. So a dihybrid cross is just where you're, instead of looking at one characteristic, for example, seed shape, which we looked at in the last example, a dihybrid cross, so di meaning two, this is a type of cross that we're going to do where we're looking at two characteristics. So we're looking at two characteristics. And so we're going to be using, for those characteristics, two letters. So we have big A, little a, but also big B, little b. So with 4 potential gametes, you're going to contribute, as this organism here, where we're only focus firing on 2 characteristics, you could make a gamete that contributes these two, big A, big B, you can create a gamete that contributes a big A and a little b. You can make a gamete that contributes a little a and also a big B, and then you could also make a gamete that contributes a little a and a little b. And so this would probably help better explain the law of independent assortment, as we'll see when we do the Punnett square. So different alleles will assort into gametes independently of one another. So kind of like I mentioned when we talked about meiosis, law of independent assortment isn't only a description of the movement of your chromosomes independently of one another. What's actually happening is your alleles themselves are what independently assort. All right, so F1 generation. So if we're dealing with an F1 generation, big R, big Y, little y, it's going to have the potential to produce 4 different gametes for that genotype. So this is the genotype for an organism. So it can make a big R, big Y, big R, little r, big Y, and a little r, little y. So far we've talked about seed shape, and if you remember, the big R was round, and then little r was wrinkled. In this case, the Y, that's just going to represent color. And so I think yellow is, the big Y is going to be the dominant. So big Y is yellow, and then little y is green. Looks like little y is green, and it's the recessive allele. So here we have a pure breeding round and yellow seeded plant. So the only potential gametes that it's going to contribute are going to be big R and big Y. And then you have a true bred wrinkled green seeded plant. So little r for the recessive and little y for the recessive green. So little r for the recessive wrinkled and little y for the recessive green. And so the only type of gamete that they're going to produce are little r, little y. So when those two cross, what you get is a heterozygote for both traits. So your first filial generation is what you get, and it's going to be big R, big Y, little y. And when we cross-pollinate, or when we pollinate self, so self-pollination, you can make 4 potential 2-letter combinations using all of these letters. So you could have a big R, big Y. Here you have a big R, big Y. You have a big R, little y, and here's a big R, little y. You have a little r, big Y, little r, big Y. You have a little r, little y, little r, little y. So it's cross-pollinated with itself. And then kind of like with the Punnett square, or for a monohybrid cross, you're doing the same thing, but you're just doing it with 2 letters. So now you have, big R, big Y, big R, big Y. What's the cross between a big R, big Y and a big R, big Y? Big R, big R, big Y, big Y. And so here's a monohybrid cross showing quite a few more potential offspring, because instead of looking at only 1 trait, you're actually looking at 2 traits. So the F2 generation, here it shows the phenotypic distribution right here. So, you have 9 yellow round, right? So you have 1, 2, 3, 4, 5, 6, 7, 8, 9 potentials of yellow and round. And then you also have, 3 potentials for yellow and wrinkled. So yellow, wrinkled, 1, 2, 3. And so this is the phenotypic ratio here. If we were to do the genotypic ratio, well, we would just be counting, how many big R, big Y, big Ys do we have? How many big R, big Ys do we have? And so on. So a pedigree is just a diagram of family history that uses standardized symbols. A pedigree will typically show the relationship between family members, and it'll indicate which individuals have certain genetic, pathogenic variants or traits or diseases within a family. So here you have, we could say that the shaded in box, or so the shaded in shapes are going to be, let's say, someone that's affected by some kind of disease. And the squares, we'll say that that's male, and the circles are female, for example. So here you have, if we're looking at This would be the grandchildren. This would be the children, and this would be the grandparents here. And so if you look here, you have two affected individuals, two people that have some kind of disease. They have offspring, and they produce two affected individuals and one unaffected individual. And that unaffected male mates with, an unaffected female, and then they produce, three offspring that are free of whatever disease. So in this case, you have 3 and 4. This is an affected male that mated with an unaffected female. They had two children, and what happened was they kind of extracted themselves from whatever disease by just mating with someone else that does not have the disease. So this is just an example of a pedigree.